So the next topic is pharmacophore analysis. So if you look at these are the various stages of drug discovery process, drug design and development. So identification of disease, identification of drug target, so establishing the testing procedure like biochemical assays, cell line assays, finding lead molecule through that, then doing the structure activity relationship. So while doing the structure activity relationship, it is very essential to identify the pharmacophore, right? So let me define what is meant by pharmacophore. So pharmacophore is the atoms or group of atoms which are commonly present in the active drug-like molecules that contribute for the biological activity. Okay, before that, so far whatever methods we have discussed like X-ray crystallography for structural determination, molecular docking, de novo ligand design. So all this comes under structure-based drug design. Okay, so this is the first ligand-based drug designing we are seeing. That is pharmacophore, right? Again, I define what is meant by pharmacophore. Atom or group of atoms which are commonly present in all the active molecules. If you remember, the main difference between structure-based drug designing and ligand-based drug designing is that in case of ligand-based drug designing, so not necessary we should know the 3D structure of the drug target. So we need set of ligand molecules with known biological activity. So for example, these are the set of known ligand molecules, set of ligand molecules with known biological activity. Say for example, these are the molecules which inhibits COX, or these are the molecules, yeah, which inhibits COX. Okay, cyclooxygenase. But we do not know. Assume that we do not know what is the 3D structure of the drug target. That is not actually needed for ligand-based drug design, right? Since all these molecules, set of molecules which inhibit cyclooxygenase, reduces the fever or reduces the pain, whatever it is, right? So if you compare this lead molecule, all the lead molecules, if you compare the structure of all the lead molecules which act on COX to reduce the pain, so there will be some feature that is common, right? And that feature, if that feature contribute for the biological activity of that molecule, then that is what is called as the pharmacophore. Again, one last time I repeat, what is pharmacophore? Atom or group of atoms commonly present in all the active ligand molecule acting on same target and responsible for the biological activity of those molecules. That is the pharmacophore, right? So in simple words, we can say that this pharmacophore elements they basically define the important group involved in binding. So we know that the functional group which are participating in binding, they are related to the biological activity. Okay, so we say that as the actives, the functional groups which are interacting with the active site of the protein molecule, or which are important for the binding of the drug molecule to the binding pocket of the protein molecule. Uh, so there is a connection between the binding and the biological activity. So another thing is that pharmacophore is not just finding out which atom or group of atom is important for the biological activity. What is the spatial relationship between those binding atom? That also we study the relative position of the binding group. Okay, that also we explore in the uh, analysis of pharmacophore and um, let me start with the 2D pharmacophore analysis. Slowly we will move to the 3D one, right? 3D is the advanced, more reliable one comparing to the 2D pharmacophore. Was but simple. We have to start with the 2D pharmacophore just for understanding the concept of the 3D pharmacophore, right? So what is this 2D pharmacophore? It defines the minimum skeleton which connects the important binding group, right? So I'm going to give some example, right? So if you take this, the morphine structure of morphine. So I hope that you know what is the biological activity of morphine. It's a very strong painkiller. So even today, it is in use, very strong painkiller. So it has analgesic activity, also some narcotic effects related to this morphine. So which means that there are different biological activity for that. So if we take this analgesic activity, pain-killing activity of this morphine, the structure of morphine, right? So we know that this structure is actually a complex structure. An interesting thing is that this molecule is derived from some natural source. So if we look at what are the various functional groups which are present, so here we have NME. There is a phenolic OH group, another OH group, aromatic OH group. There is a ether here, and so here we have aromatic ring system. So here we have another ring system. Here we have another ring system. Here we have another ring system, right? So these are the various component functional groups of this morphine. So we know that this is the structure of morphine. So it is having very good pain-killing activity. So whether the entire molecule is responsible for its pain-killing activity, or some of the functional group which are present in this morphine is that responsible for biological activity. If you do any kind of study, right? So that is what is actually called as the pharmacophore analysis, right? So I'm going to show another version of morphine, right? So here some groups which are highlighted in yellow color, right? So those are the important groups for its analgesic activity. Again, I repeat, morphine is having different activity. Narcotic activity is also activity exhibited by morphine. So if we think of analgesic activity, so the groups which are highlighted here as the red color, the OH group here, the benzene ring here, and the nitrogen here. So those are the groups which are important for its analgesic activity, right? So if we make a connection between only the most important functional group responsible for the analgesic activity, right? So then that part, okay, along with that connection, this is what is called as the 2D pharmacophore of the morphine. So we can say this is the 2D pharmacophore of opioid, right? So which is having the analgesic activity. So this skeleton is what is called as. So three important groups essential for its activity: OH, phenyl group, nitrogen, and the connectivity here, right? So it dictate how the three important functional groups are connected. So this probably we can say 2D pharmacophore of the morphine. So far, is it understandable?
Yes, sir.
Yeah. So moving further, here we have other derivatives of morphine. So if you look at the structure of morphine and metazokine, so we can see that there is some structural similarity. Okay. So if you similarly if you look at the morphine and levorphanol, so there is a structural similarity. So in the previous slide, I have shown that which functional group is important and the skeleton of the important functional group. So if you remember this, okay, if you check whether the same group at the same position is present in the derivatives of morphine, okay, probably we can say that definitely morphine is having that same group here. It goes in this way. And if you take metazokine, it also goes here with the same kind of connection here. Okay, so here if you take levorphanol, that also has the same skeleton. The skeleton is there, right? So which dictate that all the three derivatives, all the two derivatives of morphine and its two derivative, metazokine and levorphanol, so they have the same pharmacophore feature. Do you agree? At the same position. Do you agree?
Yes, sir.
So the three form of four element is there, one, two, and three at the same position. Okay, so we can see here at the same position we have the same three functional group, right? So it dictates that the form of four is preserved in metazokine and levorphanol. So then we can expect to some extent that the property of morphine could be probably exhibited by metazocaine and levorphenol based on the same pharmacophore feature is exhibited present in metazocaine and levorphenol. So that is the basics of Pharmacophore. I hope that is understandable, right? So here you can see the red color highlighted Pharmacophore element along with the skeleton, the functional groups joined together through the yellow color line, right? So that is the 2D Pharmacophore. Whatever so far I have explained is an example for 2D Pharmacophore. Moving on further, right? So we are interested on 3D Pharmacophore because that is the advanced thing. So this 3D Pharmacophore, it defines the relative position in space of important binding group. Here, skeleton is shown, but it is not in three-dimensional fashion, right? So it's a 2D fashion. So actually, what we are seeing here is the 2D representation of all the molecules, not the 3D representation, right? So it's not on the 3D space, right? So for considering 2D Pharmacophore, the 2D relationship is fine. Okay, no issues. It will give very good, clear visual inspection whether the same Pharmacophore is exhibited or not, right? But what we are interested, right? So whether that position is retained at the same three-dimensional space or not, that is very for having the very similar biological activity by the derivatives. So that is what we normally study in the 3D Pharmacophore, right? So defines the relative position, whether the Pharmacophore elements are present in the same three-dimensional space, whether the interacting binding groups of the drug molecule, which are crucial, are present in the same position in the three-dimensional space or not. Right? That is what we normally study. Um, So here, this is the 2D representation. So here, this is a 3D representation of the same molecule. Still, we are viewing the molecule in two-dimensional space, but you can see now the complexity of the molecule is in 3D space. It is it's highly complex, right? So what we study in 3D Pharmacophore is that whether the important functional group, what we discussed as this OH, this phenyl ring, and this N, okay, whether they present in the same position in the three-dimensional space or not. That is what we normally study. So here this phenyl ring, and here we have the oxygen atom, and here we have the nitrogen. Okay, so we can produce the xyz coordinate for this molecule, and we came to know what is the xyz position for this, what is the xyz position for this AR, what is the xyz position for this N, and we can compare that with the derivative of morphine, whether the same oxygen at the same atomic coordinate, whether the aromatic ring is present at the same atomic coordinate, and the nitrogen is it present the same atomic coordinate, either using the xyz matrix or z matrix, right? So mostly we prefer to go with the xyz matrix. Here, we have drawn the dotted line, which makes connection between the oxygen aromatic ring and the nitrogen. So now if we hide the molecule for simplicity, we can clearly see that there is a spatial relationship between the oxygen, AR, nitrogen. So these are called as the Pharmacophore element. We map the Pharmacophore element. So Pharmacophore element is the atom that is important for the biological activity. So here we trapped the spatial relationship in terms of distance. We can measure what is the distance between the oxygen to AR, AR to nitrogen, nitrogen to oxygen. Any combination we can measure. Similarly, there exists angle, right? Since it is in 3D space, so since we have three component O, A, R, N, so there exists angle. There is an angle exhibited between the three atoms, 3 points, which are given here: 18.5 degree, 150, and 11.3 degree, right? So we can say that this is the 3D pharmacophore of morphine. This is the 3D pharmacophore of morphine. So if any other molecule has the same oxygen atom at this position, same aromatic ring at this position, and the same nitrogen at this position, with the same spatial relationship, in the sense that the distance between O to AR is 2.798, AR to NS 4.534, and O to NS 7.098, and the angle between them is 18.5, 15 point, sorry, 150 and 11.3. Then we can say that both the molecules, say morphine and the another molecule X, they share the same pharmacophore feature at the same position, three-dimensional position. So probably it is better to look into the biological activity of this molecule X. Okay, there are more chances that the X may have the similar property, biological activity like the morphine. That is the basic idea of the pharmacophore analysis. Okay, so here comes the real topic. So how many steps basically we have for doing pharmacophore analysis? We have 4 steps of doing pharmacophore analysis. So we're going to see what are the 4 steps and what will be the final output from the pharmacophore analysis, right? The very first step is data collection, right? So what is meant by data collection? Say for example, you are interested on designing a new inhibitor for cyclooxygenase COX. So repeatedly I am using this COX as an example because we have seen the detailed molecular mechanism of action of drugs which are acting on COX. Okay, COX, prostaglandin, how that is related to pain killing activity, right? So that's why I'm using this example again and again, right? So protein again and again. So if we consider we are interested on designing new inhibitor for COX, more potential inhibitor of COX, novel inhibitor of COX, whatever it is, right? So let us assume, right? So not in real, let us assume that we do not know what is the 3D structure of the drug target. But we know what is the 3D structure. Assume that we do not know what is the 3D structure of the cyclooxygenase. Right? But we know set of ligand molecules that inhibits the COX. So these are the set of ligand molecules. Through the biological assays, I came to know that these are the molecules which perfectly inhibits the cyclooxygenase at say nanomolar, micromolar concentration. Very good activity for the lead molecules, right? So our intention is that we had to improvise the biological activity. We had to find some other molecule having better biological activity, right? So in any way we can use this pharmacophore analysis. First thing is we had to identify what is the pharmacophore element, common feature that is present. All the known inhibitor that inhibit cyclooxygenase, which are having good activity at nanomolar to millimolar concentration, right? So either you can do this experimentally or we can collect this information from the literature. There may be already a lot of literature in which they have shared the information, research detail about the known inhibitor of cyclooxygenase and what is the biological activity. Okay, most bioinformaticians will start from the literature review, capturing from the literature. They won't do the experiment, right? So they do the computational part, not the wet lab analysis. But if you are skilled in wet lab, if you have money for funding for the project, then go for finding take set of lead molecules, find which compounds are inhibiting COX, find out what is the biological activity of those, right? So now what we know is we know set of lead molecules that inhibits cyclooxygenase. We know what is the structure of those molecules and what is the biological activity of those molecule. We are no way related concerned about the 3D structure of the drug target. This is what we have. Okay, this piece of information you can very easily collect from the literature. There may be many literature in which they have given series of derivatives of some drug-like molecules with known biological activity of some target. So we have to collect this information from the literature. So that's the first step. Collect set of high affinity ligand molecule for target receptor. Right? So we have to collect the high affinity ligand molecule. So choosing the data set, while choosing the data set, we have to follow certain conditions, or we have to give importance for certain things called as consideration. Okay, so what are the consideration? The first consideration is that the molecule should have diverse structural framework. While choosing the data set, either from the literature or from your own laboratory, you have to ensure that the molecules are having high structural diversity. You may have a question that why the molecule should have high structural diversity, because our intention is finding out that which feature is important for the biological activity computationally. We're not going to do any structure activity relationship practically, right? Computationally, without any wet lab, we had identified. If all the molecules looks very similar, then the algorithm will understand that all the features are important for the biological activity. If all the molecules has the same biological activity, but if they are structurally diverse, then it will be easy for the algorithm to pick up that which feature is important for the biological activity. That is why the molecules should have diverse structural framework. So second thing is that they should have less torsional degrees of freedom. So probably I have some example in the upcoming slide. So the reason as of now is that if the molecule has less torsional degrees of freedom, so this torsional degrees of freedom I discussed while discussing about the docking algorithm, if you remember. So there are basically 6 degrees of freedom, 3 for translation and 3 for rotation, right? So and many for the torsional degree of freedom. So torsional degree of freedom is nothing but the rotational bond, the number of rotational bonds. Okay, depending upon the number of rotational bond, the number of conformation will increase. It can reach exponential, right? So if the molecule has many number of rotatable bond, the torsional degrees of freedom will be more. So to avoid that, we are ensure that the data set what we are choosing or having less torsional degrees of freedom. So, and as we just do the data collection from the literature, okay, we have to ensure that the data that we collect is of high quality. There are many ways by which we can find the biological activity of the drug-like molecule, starting from very simple assays to very complex assay. Some assays will give very accurate binding result, activity result. So, for example, a radio ligand binding assay, a set of experiment which comes under radio ligand binding assay. So, these are the assays which will prove that the molecule what is in your data set, it's actually binding to the cox. Okay, so we label the ligand molecule with some tag. So we finally measure whether we finally ensure whether the drug molecule bind to the target or not using that label, using fluorescence, emission of fluorescence or any method, right? So radio ligand labeling is one of the very high quality assay. So we ensure that the data we collect used any of this kind of radio label like ligand assay, preferably from the same laboratory. This is whenever we do the data collection, right? So preferably if you take from the same laboratory, then it has added advantage. Okay. Reason is that if you take one data from Indian scientist, another one from another country, right? So the experimental procedure may change, the pressure, temperature may change. That may have some influence on the binding assay data. So in order to avoid that, we should prefer from the same laboratory. Those are the considerations. So this is a data set, right? So here, if I ask a question whether the structures given in this image, they are structurally diverse or not, so they're not diverse. Right? So we should not choose the data set like this. The reason is that the algorithm will get confused. All the structure looks very similar, right? So if you ask the algorithm that which functional group is important, common, present in all the molecules, then the algorithm may end up like it will tell that all the functional groups which are present are important for the biological activity. So the structure should be slightly diverse in nature, right? So that the first point we have to keep in mind. A second point, what I said is torsional degrees of freedom. So while choosing the molecule, we have to ensure that the molecule is having some kind of rigidity. It's not completely flexible. So example for rigid molecule is the codeine. Even if you take morphine, right? So that is example for rigid molecule, right? So codeine is another analog of morphine. So here normally you will be having OH in morphine. The hydrogen is replaced by methyl. That's the only difference between morphine and codeine. So this molecule is rigid in nature because the number of rotatable bond is rigid. The torsional degrees of freedom is less. But if you take phenoxidil, is another drug molecule. So this is highly flexible molecule. How do you say it is highly flexible? Because there are more and more number of rotatable bonds which are present. So if any molecule has more number of rotatable bonds, then obviously it can be present in many conformation, right? So the molecule is highly flexible. So if the molecule is highly flexible, then it will be very difficult for you to align the molecule. That's the next step what we are going to do, right? So during the alignment process, it's going to give some problem. You may not have very good software for aligning the molecule to find out the bioactive conformation. That is one reason that why we should prefer to go with the slightly rigid molecule than the flexible molecule. That is about the first step. So coming to the second step, right? The first step, we just collected set of ligand molecules with known biological activity with certain consideration. So what's the second step? For all the molecules in the data set, in the second step, we have to do the exhaustive conformational analysis. We have to do exhaustive conformational analysis. So you may have a question, what is this exhaustive conformation analysis? I hope that BTech students, at least you know what is meant by conformational analysis, right? So if we take this molecule as example, so this is ethane, right? Just for the MSc students, I don't know whether any MSc students joined or not. So anyway, so if you take this molecule as example, so here, so I label this one first carbon, second carbon, third carbon, first, second, and third carbon. So this has a carbon-carbon single bond, which is a rotatable bond. So if we rotate, the position of this three atom will get changed. The position will get changed. Two will come to first position, first will go to the third position, and so on. Right. So we can generate n number of conformation. Okay. So the number of conformation depends on the degree of rotation. Okay, depending upon the degree of rotation, I can generate n number of conformation. That is what is called as the conformational analysis, right? So what is the exhaustive conformational analysis? So if you perform complete conformational analysis for this molecule with all possible angle of rotation, okay, all possible angle of rotation should be given. If you do such conformation analysis, that is what is called as the exhaustive conformation analysis. So if we take another example as methane, ethane, propane, so CH3, CH2, CH3. So now the number of rotatable bond increased. In the previous case, we had only one number of rotatable bond. In this case, we have two number of rotatable bond. So we can rotate with respect to this bond, and with respect to this bond, we can rotate simultaneously. Okay, so if you generate all possible conformation with all possible rotational angle of degree, then that is what is called as the exhaustive conformational analysis. So we have to do this exhaustive conformational analysis for all the compounds in the data set. So for example, in the first step, you have selected 25 compounds as the data set. For all the 25 molecules, we have to do the exhaustive conformational analysis. So if you do the exhaustive conformational analysis, all possible conformation of the 25 molecules will be identified with the corresponding energy. So why do we do this conformational analysis? Because each conformation of the molecule will have certain energy. Okay, all the conformations will not have same. So you might heard about all those terms. I'm not going in deep. Skew conformation, eclipse conformation, staggered conformation. Okay, these are some terms you might heard, right? So different conformation of the molecule is associated with different energy. So once if you do the exhaustive conformation analysis, all possible conformation and the corresponding energy can be identified. So moving on further, first step is we collected the data set with some consideration. For all the compounds in the data set, we performed the exhaustive conformation analysis. So we know all possible conformation and the corresponding energy in the second step. That's what I explained so far, right? Some images for you to understand the concept clearly, right? So a person, different position. If you assume this as the molecule in the data set, we can say that the same the lead molecule in the data set, any one lead molecule is present in different conformation. So this person is present in different conformation. So each conformation is associated with different energy. So the person is lying here, the person is standing here, person is sitting here, right? So the person is upside down here, different conformation, right? Each conformation is associated with different energy. So while lying he is having low energy, while sitting also he is having low energy, while standing also he is having low energy, while present in this conformation, right? So upside down with one hand he is having very high energy. So this is another high energy state, right? So it will be difficult for the person to be present in this state for a long time. This is medium energy. For some period of time, he can be present in this conformation. He can also be present in this conformation for some time. Another conformation, he can present long time, low energy, right? So which means that each conformation of the same molecule is associated with different energy. So we completed the exhaustive conformation analysis, and we have to identify that which conformations are low energy conformation. So in this is a low energy conformation. This is a low energy conformation. This is another low energy conformation. This is another low energy conformation. So these are the conformations we are interested on. We are not interested on the medium energy or the high energy conformation. The reason for picking the low energy conformation in step 2 through exhaustive conformation analysis is that the bioactive conformation, okay, mostly they used to be any of this low energy conformation. Again, I repeat, why do we discard this medium energy and the high energy conformation? Most of the cases, the bioactive conformation of the drug molecule. The bioactive conformation is the conformation in which the drug molecule can bind to the target to produce the desired biological response. That conformation most of the time used to be any of this low energy conformation. Going further, we are in step 3. Literally speaking, step 3 is the last step of pharmacophore analysis. But we normally used to say that there are 4 steps, right? So let me finish step 3, and you can understand why this is the last step of pharmacophore analysis. While going to step 4, you will understand that why that step is optional and how important is that step, right? So what is the third step? Third step is the computationally very expensive step. Okay, so this is a place where the companies are tricky in writing the algorithm. So what we normally do is that we take the data set. Okay, end of second step we have the data set compound, and for each compound we have various conformation, all possible conformation, and out of that we picked which conformations are low energy conformation. So that is what we have for this compound 1. Like that we have for all other compounds. This is for the second compound. So this is for the third compound. Like that, whatever is the number of compounds in the data set, all those will have the low energy conformations. There is no one compound, right? One compound, now different many conformations are representing one compound. These are many conformations of the first molecule, different conformations, low energy conformation of second, different low energy conformations of third, and so on, right? So what we have to do is that we have to do the molecular superimposition. Again, molecular superimposition is not new concept for you, right? So we discussed what is molecular superimposition while discussing about the chirality, the importance of stereochemistry in drug design. We discussed non-superimposable mirror image. That time I told you how to superimpose, right? So you cannot superimpose the left and right hand. Okay, that is what you have to, simple example for understanding what is in that way you have to superimpose one molecule over the another molecule, right? So we have to take all the low energy conformation of all the molecules in the data set, and we have to perform this molecular superimposition. Okay, so why do we do this molecular superimposition? So this is the principle used for finding the bioactive conformation. So once if you complete step 3, will tell you which conformation is the bioactive conformation. This is the actual question which I asked you, right? So how will you find out the bioactive conformation? Since we do not know what is the 3D structure of the drug target, okay, so we rely on the computational methods for finding out the bioactive conformation that is identified through the molecular superimposition, right? So let me give one example. You will clearly understand what is molecular superimposition, how we do that. So here we have three molecules, say molecule number 8, 13, and 22. So those are the three molecules, three different molecules in the data set. Okay, so these are three different molecules of the data set. So this is molecule 8. Uh, I write here, this is molecule 8, and this is molecule 13, and this is molecule 22. So like that, you may have many number of molecules. Just for example, we are taking three molecules from the data set for explanation, right? We had actually do for all the molecules, right? So this 8 will be present in different conformation. Say all are low energy conformation. These are the low energy conformation of the molecule 13. So these are the low energy conformation of molecule 22 and so on. So say this is one molecule, one conformation of molecule 8. This is one conformation of molecule 13, and this is one conformation of molecule 22. Hope now that is clear, right? So one conformation of molecule 8, 13, and 22. So if we look at this by simple visual, we can say that there are some functional group which are commonly present in all the molecules. So definitely we can say that this NH2 group is commonly present in all the molecules 8, 13, 22. Right? So maybe in all other molecules also. Just for example, three molecules we are taken. So this COOH group is present, all the molecules. And this benzene with phenyl ring is present, all the molecules. If you try to superimpose this molecule, keeping one molecule over another, right? So what will happen that perfectly we can align this group with this group with this. Do you agree? Just move the molecule and keep this group over this. Keep this over this. All the benzene with F will be superimposed. So if you do so, will this group get superimposed or not? Because we are thinking to solve this problem in through two-dimensional way, right? So we have to think this to solve in three-dimensional way, right? So what actually does is that the algorithm will identify that which functional group is commonly present in all the molecules given in the data set. Again, I repeat, which functional group is commonly present in all the molecules in the data set. Secondly, it will try to superimpose each conformation of the molecule in all possible ways. The first conformation of this superimposed with first of this, superimposed with first of this, superimposed with first of this, first of this, first of. So first of this molecule superimposed with the second of this, second of this, second of this, and so on. So in all possible conformation, we have to superimpose all the molecules. Okay, so in which combination most of the functional groups are superimposed three-dimensionally, and that conformation is assumed as the bioactive conformation. Maybe slightly confusing for you. Just look at this image in the bottom, you can easily understand. So this is the phenyl ring with F. Okay, so the phenyl ring with F is present here in all the three molecules. So this COOH group, this NH2 group is superimposed here, all the three compounds, and the COOH is superimposed all the three compounds here. So do you agree that all the three functional groups are superimposed?
Yes, sir.
Okay, so now the conformation in which most of the functional group superimposed like this, that conformation is what is called as the bioactive conformation. Is that understandable? That is what is called as the bioactive conformation. So now we can say that this conformation is the bioactive conformation of this data set. Is that understandable? So now we can say that these are the pharmacophore elements 1, 2, and 3. And there is a spatial relationship between the pharmacophore element. Okay, that is what we actually studied in the 3D pharmacophore